Hello, this is Dibyendu and I am a PhD student at the University of Calcutta, and today I am going to present our work identifying cell cluster specific gene interactions for single cell transcriptomes using association rule mining. So let me go through the problem statement first. Whenever we work with single cell ionophilic data, we often need to find out statistically significant genes across cell clusters or differentially expressed genes across different cellular conditions. Now, these kind of analyses are often based on the presence or absence of marker genes. Now, the problem here is that cells, even though they might have similar gene expression profiles, may not always represent the same transcriptional state. So finding out differentially expressed genes might not be sufficient enough to uncover the molecular activities within a particular cell type. So what we are suggesting here is that we need to find out major gene-gene interactions within a particular cell cluster, and analyzing these gene-gene interactions will lead us to identify key regulatory hubs or genes that are driving other genes or maybe being driven by other genes, and eventually we will be able to find out the molecular signatures of this particular cell cluster or understand the underlying molecular activities within this cell cluster. So this was the motivation of our work, and hence we have proposed an association rule mining based framework to solve this problem. Now let me go through the basics of association rule mining. Association rule mining, or ARM, was initially proposed for market basket analysis, where we used to analyze the association between two items or products, and these kind of analysis helped us to understand customer buying patterns and make target promotions for them. The strength of an association rule was often found out by different parameters like support, confidence, and lift. So, in this work, what we have done is, instead of items, we have considered genes, and we have tried to find out major associations between these genes within a particular cell cluster of interest. So let me mention here that association rule mining was earlier used on bulk RNA-seq data as well as microarray data. And now we have proposed association rule mining framework for single cell RNA-seq data, where we can identify major gene-gene interactions within a particular cell cluster. And after identifying the major gene-gene interactions, we have tried to build a network out of these top gene-gene associations and identify key regulatory hubs or genes within that particular cell cluster. Uh, After that, we have tried to validate the genes that have been identified as key regulators by our method against benchmark methods like FURAT. We have also validated these set of genes against another reference-based cell type database, DB, and we have found that these genes are common when compared against the top 10 positive markers identified by FURAT, and these genes have also been identified as canonical markers when verified against this database DB. So finally, what we have done is we have taken these hub genes and their associated neighbors, and we have formed a gene list. And for each of these hub genes, these gene lists have been further assessed for pathway enrichment using WebGSORT. So this is the overall workflow. We have pre-processed SCINFIC data. We build a transaction dataset from that and apply association rule mining on that, and we get the network of top association rules. From this network, we try to identify the hub genes or the key regulatory genes, and for each of these hub genes, we prepare a gene list which comprises of these hub genes as well as their neighboring ones. And finally, these gene lists have been analyzed for overrepresentation based pathway enrichment analysis using WebGSORT. So we are trying to find out, we are trying to see what kind of events are running within these particular sub networks. Okay, so that is the workflow of the proposed method, and let me go through the results. So for the time being, we have just used PBMC 3K dataset, and we have analyzed NK, B, and CD8 plus T cell clusters. So these are the three cell clusters that have been analyzed, and table 1 shows the support thresholds that have been used for each of these cell clusters, the number of initial rules, and the number of rules before and after filtering. And figure 2 shows the distribution of rules, where we can see that most of the rule lengths were 4, 5, 6, 7, 8. That means most of the uh, in most of the association rules there were around 4, 5, 6, 7, 8 genes associated or interacting with each other. Now this is the kind of network that we have got from this association rule mining. So let me zoom out a little bit, and as we can see that we have identified key regulatory hubs from this network by a systematic procedure. Like for example, we can see GZMB here interacting with all these genes. We can see a hub for PRF1, uh, again GNLY, CTSW, CST7, and finally we have compared these regulatory hubs with those obtained by FURAT, and we have seen that most of the regulatory hubs identified by our method are also common with those obtained by FURAT, and these hubs also are canonical markers for different cell types as verified by the Bangalore DB database. Finally, what we have done is we have considered each of these hub genes and its associated neighbors, and we wanted to see what kind of underlying activity is going on in this sub network. So we have prepared a gene list for each of these hub genes, which comprises of these regulatory gene hubs and the associated neighbors, and we have we have assessed them for pathway enrichment. So as you can see, the GNLY network has shown enrichment of mostly post-transcriptional and translational events that are often required for the activation of antigen-pressing cells or APCs. The GZMB network has shown enrichment for translation-related, ribosomal transport-related, and intracellular transport-related pathways, which indicates its role, the primary role of this molecule GZMB or granzyme B in apoptosis and membrane necrosis. So what can we conclude from this is that the association rule mining based framework has made us find strong gene associations, and these kind of analysis leads to something beyond traditional rule based cell type discovery, which is based on the presence or absence of marker genes. Now, this kind of framework could be in future extended to analyze unannotated or rare cell clusters that may or may not express any canonical marker. So that's the conclusion from our work. So that's all from my part. I would like to thank my co-authors Dr. Vivek Das and my PhD supervisor Dr. Rajat Kumar De, and I would also like to thank the ISMB 2022 organizers. Thank you.